My topic for the presentation is types of mutations. So let us start the presentation on the types of mutation. So what is mutation? Sudden heritable changes in genetic material or character of an organism is referred to as mutation. Individuals showing this mutation are called mutants. So what is mutation? Any change in the genetic material, sequence of the chromosome, a part of chromosome, a part of DNA is called a mutation. Two main types of mutations are there, that is point mutation and the chromosomal mutation. Point mutations are also called the gene mutations. Mutations are random events. So what are the bases of classification? Mutations can be classified in various ways. So based on source, two types of mutations are there, spontaneous and induced. So what are spontaneous mutations? Mutations that occur in nature. It involves deamination and depurination. So what is depurination? As we know, purines are the nitrogenous bases. If they are removed from the sequence of nucleotide, it is called the depurination. This is sugar, this is phosphate, this is the nitrogenous base. If the guanine is removed, it is called depurination. So sugar, phosphate are remaining after the depurination. Deamination, as we know, in the cytosine there is the NH3 at the second position, and if it is removed, it is called the deamination, and cytosine is converted to the uracil. Example is UV light of sunlight causing mutation in bacteria is the example of spontaneous mutation. Next is induced mutation, mutations that are produced by use of mutagenic agents. Example is X-rays causing the mutations in cereals. Next is on the basis of direction. Two mutations are there, forward and reverse. Forward mutations are the mutations occur from the wild type allele to mutant allele. Wild type are the original type, and when the mutation occurs from the original to the mutant is called the forward mutation, and when mutation occur in the reverse direction, that is from mutant to wild allele, are known as reverse mutation. Next is on the basis of tissue. Two types of mutations are there, somatic and germinal. Somatic mutations are the mutations in the somatic tissue. For example, in asexually reproducing organisms, somatic mutations transmit from one progeny to next. And germinal mutations are the mutations that occur in gametic cells or reproductive cells in sexually reproducing organisms or species. Germinal mutations transmit to the next generation. Next is on the basis of site. Two types of mutations are there, nuclear and the cytoplasmic. A mutation in the nuclear gene is called the nuclear mutation, and mutation in the cytoplasmic gene is called the cytoplasmic mutation. Based on survival, four types of mutation are there: lethal, sublethal, subvital and vital. So what are lethal mutations? A mutation that kills the individual that carries it. Sublethal are the mutations when the mortality is more than 50% of individuals that carry mutations, and when the mortality is less than 50% of individuals that carry mutation is called subvital mutation. Vital mutations are the mutations when all mutant individuals can survive. Based on character, there are two types of mutations, morphological and biochemical. What is morphological? That is the mutation which affects on phenotypic characters, that is external characters that can be seen or observed by the normal observations are called the morphological mutations. The mutations which affect the production of biochemicals and which does not show any phenotype character are called the biochemical mutations. Next is chromosomal and gene mutations. So what are chromosomal mutations? It may involve changing the structure, loss of gain are involved. Five types exist: deletion, inversion, translocation, non-disjunction and duplication. So in this, a segment is deleted, in this is called deletion, and in this translocated, that is, it is the one chromosome, this is the another non-homologous chromosome, some part is translocated to the other. In this, duplication occur, that is A, B, C, D, E and F. F is here, but A, B, C, D is duplicated to the A, B, C, D, E. So this is called a duplication, that is A, B, C, D, E is duplicated. A, B, C, D, E, A, B, C, D, E, F. Okay. Now inversion means when segment rotates 180 degree, that is A B C D E F था यहाँ पर, वो invert हो गया and F A D C बन गया and it is called the inversion. So these mutations are the chromosomal mutations. Next we come to gene or point mutation. A gene mutation is an abnormal change in DNA. Three types of DNA or gene mutations are there: base substitutions, deletions and insertions. Base substitutions are further of two types, transition and transversion. And transversion is further of three types, silent, nonsense and missense. Other two types are deletions and insertions. Deletions and insertions are also called the frame shift mutation. Indel is the combination of insertion and deletion. In for insertion, del for deletion. Let us study in detail about these. So what are base substitution? Single base substitutions are called point mutations. Example, the point mutation of glutamine to valine. Glutamine is amino acid, valine is also amino acid. If in the sequence of polypeptide, glutamine is substituted to valine, which cause sickle cell disease. Point mutations are the most common type of mutation and there are two types. Transition is a type of base substitution. This occurs when a purine is substituted with another purine or when a pyrimidine is substituted by another pyrimidine. Second type is transversion, when a purine is substituted for a pyrimidine or a pyrimidine replaces a purine. So transition is shown here. As we know, T and C, thymine and cytosine, are the pyrimidines. If they substituted interconvertibly, means thymine is substituted by cytosine or cytosine is substituted by the thymine in a position of the codon, it is called the transition. Purine to purine, as we know, A and G, adenine and guanine are the purines, and same in the case of pyrimidine, if they substituted by each other, it is called the transition of purines. Next is transversions. Transversion में क्या हो रहा है कि T is the pyrimidine and if it is substituted by A or G, and C is also a pyrimidine, if it is substituted by G or A, it is called transversion, and vice versa for purine to pyrimidines. So these are two cases of transition and transversions. As we know, transversion is further of three types, that is silence, missense and nonsense. So this is the normal sequence: AUG for methionine, GCC for alanine, TGC for cysteine and so on. So when silent mutation occur, the third position of codon is changed, that is here GCC, and third position of the codon is substituted by T. C is substituted by T. Cytosine is substituted by the thymine base. But by this mutation, by this silent mutation, alanine remains alanine, means it is silent. Mutation तो हो रहा है but no effect, function पर कोई effect नहीं पड़ रहा है. So these kind of mutations are called the silent mutations. Next is nonsense mutation. In nonsense mutation, a stop codon occurs at the position. अगर stop codon आपका add हो जाता है, then it is called the nonsense. It will lead to the non-functioning of the protein. Missense mutation में क्या होता है? Missense mutation will change the whole amino acid, that is TGC, and if T is replaced by the G, it will lead to the amino acid changing, that is cysteine will replaced by the arginine, and this will change the whole functioning of the protein. This is the brief description, that is silent. If a base substitution occurs in the third position of a codon, there is a good chance that synonymous codon will generate it. Thus the amino acid sequence encoded by the gene is not changed and mutation is said to be silent. Next is missense, when base substitution results in a generation of codon that specifies a different amino acid and hence leads to a different polypeptide sequence. Depending of type of amino acid substitution, it is of two types, conservative and non-conservative. Conservative में क्या होता है? If structures and properties of substituted amino acids are very similar to original amino acids, that is if leucine is substituted to isoleucine. They are similar. Not similar मतलब क्या है कि थोड़ी बहुत प्रॉपर्टीज मिलती हैं उनकी आपस में, तो ऐसे म्यूटेशंस को हम बोलते हैं कंजर्वेटिव. बट इफ there is no similarity between the structure and properties, it is called the non-conservative. For example, as we learned in the previous slide, that when glutamine is replaced by the valine, in the case of sickle cell point mutation, it is called the non-conservative mutation. Nonsense mutation, when a base substitution result in a stop codon, ultimately truncating translation and most likely leading to a non-functional protein. Now GC to AT transition. In case of induced mutations, this can be done by base analogues. What are base analogues? Base analogues are the structure that are very close in structure to the nitrogenous bases. As we know, purines and pyrimidines are the nitrogenous bases, and if some structures are similar in their structures or closely related to them, these type of structures are called the base analogues. For example, one base analog widely used in research is 2-aminopurine. That is, in the purines at the second position, if amino group is attached, it is called 2-aminopurine. This analog of adenine can pair with thymine. As we know, adenine only pairs with the thymine, but in the case of 2-aminopurine, it can also mispair with the cytosine. So this will disturb all the functioning. So 2AP is incorporated into DNA. So अगर आपका ये 2AP DNA में incorporated हो गया है by pairing with thymine, later ये C के साथ भी मिसपेयर कर सकता है, that is cytosine के साथ भी ये मिसपेयर कर सकता है, that is it is called the AT to GC transition by mispairing with cytosine in subsequent replications, or reverse as GC to AT transition. So in this diagram it is shown 2AP is pairing with the thymine, but the protonated form of AP, 2AP, that is N plus and H, it will lead to the pairing of 2AP with the cytosine, and this mispairing with cytosine in subsequent replication, or reverse as GC to AT transitions. These transitions can be caused by alkylating agents like ethyl methane sulfonate, that is EMS, and nitrosoguanidine NO. Example, guanine pairs with cytosine. As we know, guanine can only pair with the cytosine, but its alkylated form can pair with thymine, while thymine pairs with adenine. For example, it is guanine, it is its alkylated form. That is, as we know, the alkylating agent EMS cause the alkylation of the guanine. So this is O6-ethyl guanine. So this is the alkylated form of the guanine that can pair with the thymine. So this is GC to AT transition. And same in the case, it is thymine. This is alkylated form of thymine. As we know, thymine can only pair with adenine, but its alkylated form can pair with guanine also. So this is the case of AT to GC transition. So it's very interesting to learn that alkylating agents can cause mispairing, and this is called mutations. Next is deletions, a mutation in which a part of chromosome or a sequence of DNA is lost during DNA replication. A deletion resulting in a frame shift results when one or more base pairs are lost from DNA. So what is deletion? If some part of chromosome or a sequence of DNA is lost, is deleted, is called a deletion. If deletion is short, it doesn't fatal. But if deletion is some large segment is deleted, it can be fatal. So deletion can be artificially induced by intercalating agents like proflavin, acridine orange, and ICR 191 by slipping between stacked nitrogen bases by double helix. This is called misalignment mutagenesis. So in this, deletion, insertion, and indel is shown. So in this case, GCC is there for alanine. If C is deleted, no effect on the sequence. Alanine remains alanine. But in the case of insertion, as GCC is there, one more C is added. Now whole sequence is changed. AUG, GCC, and one more C is added, then C is added to TG, तो CTG अलग बनेगा, GC, CAA and ACG, so whole sequence is changed. In the next slide, we will learn about detail of this insertion. Insertion and deletion, when both occur simultaneously, is called indel. In this sequence, C is added and from this position, C is deleted. From this, C is deleted and inserted to this position. So it is called indel. In for insertion, del for deletion. So insertion and deletion occurs simultaneously. So insertions, what are insertions? The insertion of additional base pairs may lead to frame shifts. Consider the following sequence of bases. As in the previous slide, we have shown AUG, AAU, ACG, GCU. That is, AUG is your start codon. Then next is asparagine, threonine, alanine. Now, assume an insertion occurs in this sequence. Let's say an A nucleotide is inserted after the start codon. अगर आपका AUG के बाद AAU है and आपका यहाँ पर एक AUG और इसमें एक और A ऐड कर दिया जाए, then complete sequence is changed. स्टार्ट के बाद एस्परजीन था यहाँ पर एंड अब यहाँ पर क्या बन जाएगा, लाइसिन, टायरोसिन, ग्लाइसिन. द होल सीक्वेंस दैट इज एडिंग ऑफ a single base will change whole sequence. When the chromosome structure is altered, this can take several forms. As in the few previous slides we have discussed about the chromosomal mutations. This is a more slide of that. This is the normal A, B, C, D, E, F, G. ठीक है. In the inversion case, what is happening here? A, B and D and E are inverted, C and F are inverted. That is, D takes the position of E, E takes the position of D, C takes the position of D. So it is called inversion. Duplication case में क्या हो रहा है कि D and D duplicate हो गए हैं. Deletion case में क्या हो रहा है, C जीन आपका delete हो गया है. Insertion case में क्या हो रहा है कि एक L जीन आपकी add हो गई है तो insert हो गई है. Translocation में क्या हो रहा है, two strands are there, A, B, C, D, E, F and second is L, M and O, P, Q. Translocation में क्या हुआ कि दोनों strands ने आपस में translocate किया, that is A, B, C and O, P, Q. O, P, Q is translocated to the A, B, C strand and D, E, F translocated to L, M, N strand. So this is the translocation. So these are some types of chromosomal mutations. Next are suppressor mutations. This is the unique event that occurs at a site that is distinct from the site of original mutation. Thus, an individual organism with a suppressor mutation is a double mutant. For example, if an individual is there and some mutation occurs in the individual and one more mutation is occur at some different site of the original mutation, this kind of mutation is called the suppressor mutation, possessing both the original mutation and suppressor mutation, but exhibiting the phenotype of an unmutated wild type. There are two classes of suppressor mutations, intragenic and intergenic. So what are intragenic? As the name suggests, intragenic suppressor is in the same gene. If mutation is in the same gene, that is original mutation and suppressor mutation, both are in the same gene. This type of mutation is called intragenic suppressor mutation. The suppressor may change a second nucleotide in the same codon that was altered by the original mutation, producing a codon that specifies the same amino acid as the original unmutated codon. Such suppressors are called revertants, which are of two types, true and partial. So what are true revertants? True revertants restore the exact original amino acid, while the partial revertants replace original amino acid with a chemically similar, not the exactly same but chemically similar. In the next diagram it is shown that this is true revertants. In this it is the normal isoleucine, leucine, histidine. When original missense mutation is happened, leucine is converted to the phenylalanine by the replacement of bases from C to T, C to U. Leucine is converted to the phenylalanine. And when some other mutation occurs, that is suppressor mutation occur, reversion occurs here, that is phenylalanine further converted to the leucine and leucine So in this case, what is happening, that leucine is converted back to leucine and leucine. So this type of revertants is called true revertant. Partial revertants, leucine in the first original missense mutation is converted to phenylalanine, but in the second case it is converted by partial reversal into the isoleucine. Isoleucine is not same as the leucine, but it is closely related to the leucine. So this type of revertant is called the partial revertant. So these two cases are of the true revertants and the partial revertants. Second case is compensatory mutations. That is, a second mutation offsets the damage created by the first mutation. As we know, the second mutation compensates the effect created by the first mutation. Next and last is intragenic suppressor. Last but not the least is intragenic suppressor mutation. Intragenic, in contrast, occur in a gene that is different from the one bearing the original mutation. So intragenic, in this slide intragenic is not there, intergenic is there. So intragenic we have already read. This is intergenic. Intergenic, that is within the two genes. So this is the with the wild type sequence, transcription, translation, and in the last leucine is incorporated into the protein, that is full length functional protein. In this, a base substitution at one site produces a premature codon. यहाँ पर क्या हुआ, एक बेस सब्स्टिट्यूट हो गया एंड यहाँ पर आपका TTC और AAC था, यहाँ पर क्या हुआ, सब्स्टिट्यूट हो गया एंड अ स्टॉप कोडॉन इज एडेड which halt protein synthesis, resulting in a non-functional protein, that is termination of translation, shortened. जहाँ पर full length था आपका, यहाँ पर shortened हो गया है, non-function हो गया है प्रोटीन. यहाँ पर क्या हुआ कि साइट 1 है, ये साइट 2 है. साइट 1 पर आपका फर्स्ट म्यूटेशन हुआ था एंड साइट 2 इज अ जीन एनकोडिंग टायरोसिन tRNA. सेकेंड बेस सब्स्टिट्यूशन म्यूटेशन, यहाँ पर सेकेंड जीन है ये आपकी. नॉर्मल transcription produces a tRNA with an anticodon AUA which would pair with the tyrosine codon UAU in the translation. If the base substitution introduces the incorrect base G, the resultant mutant tRNA has anticodon AUC instead of AUE. As we know, for AUC, which can pair with the stop codon UAG and translation continues, past the stop codon, tyrosine is incorporated into the protein and it is the second mutation. So that occur in a gene that is different from the one bearing the original mutation. So this is the case of intergenic suppressor. Thank you.